My name is Meraj Khan. I'm from Toronto, Canada. First of all, I would like to pay thanks and my gratitude to Bits and Bio, and definitely for this wonderful opportunity to share what we are doing, why we are doing, and how that is going to impact our community. So, I'm a co-founder, but I'm not the one. I have a huge team and collaboration behind me. And the DigiBiomics is not a organization or a company. It's a vision, and in that vision, what we are trying to provide early disease diagnosis and personalized treatment. And under this, Pulmodeep is our flagship product, where we are very close to deploy and build to the market. And the background of Pulmodeep is just not an idea, rather this is this is the impact or the outcome of hardcore 2 decades research. I am a research scientist where I spent my PhD and postdoctoral research in lung infection and inflammation. And when we do research in that's called a bench side to the bedside, the huge problem or the gap, what I see is the translation. There's lots of research, but to translate for the patient care, that takes decades and decades. So that's what this idea or this work is came in, and we started working on Pulmodeep, how efficiently we can diagnose the problem, and based on the diagnosis we can provide the treatment, focusing on lung anomalies. So can you go to the next slide? So we see here that lung diseases, lung anomalies, they are kind of a silent epidemic. And you won't believe and that every year around 6 million deaths happen because of the COPD, lung cancer, tuberculosis, pneumonia, and many more. And if you see here in the pie chart, and in that pie chart, like these are the 3 key diseases like COPD, lung cancer, pneumonia, and tuberculosis that contributes the mortality rate in huge globally. And 6 million deaths every year, that is something, it's not a kind of just a number, it's a kind of huge problem. And I can say as title, it's a silent epidemic. Next slide. So the next part is, what are the reason for that? What are the challenges? And based on understanding the reason and challenges, definitely we can find opportunities. So the key factor is misdiagnosis. And if I reflect, misdiagnosis means what? Misdiagnosis means death, in fact. Let's say there is unfortunately, someone has lung cancer and he or she diagnosed as COPD, it's done. So, and even in top-notch research medical colleges and hospitals here in even North America, we see there's a 20 to 30% misdiagnosis happen, whether it's lung cancer diagnosed as COPD or COPD lung cancer, lung fibrosis, some other disease, and vice versa. That's the one problem. The second problem is late detection and variability in clinical expert. This is something which I want to give some time. So we are not saying that the pulmonologist or radiologist, they do not have expertise. They have expertise for sure, but human expertise is confined to personal experiences. Let's say I'm a doctor based in Toronto, and I see a particular set of patient, particular set of problem, and I'm biased to that. But my colleague who is in not in US, Washington, or a colleague in Middle East or in India, they have their own expertise. So that is a variability which causes a huge disease delay. And the technology which we have, the data analytics and AI, that is something which is going to minimize this variability. So having these challenges and opportunities, we brought this Pulmodeep idea, and we work on it. And our initial findings were very amazing. And by using the data analytics, leveraging different sets of data that I'm going to explain in the next slide, we were very effectively diagnose this problem. So here the objective is how we can what is our need currently, and how we can deploy or apply or this computational power, analytics power, algorithm, and at the end, so-called AI with different models to effectively early diagnose the lung anomalies. And lung anomalies means there are many lung diseases. I do not need to repeat all those. Along with that, how we can improve the recovery process and prevent the disease decline. Means sometimes what happens if the treatment is late, is taking time in terms of triaging and finding the right specialties, the disease get exacerbated in even not only in the lung, even in many other diseases. So that we have to reduce. So this is actually not just a tool. This is a unmet need for our community to have something which can precisely early diagnose lung problem, triage to the right specialty, and that patient get a right treatment. Next slide, please. So coming to the methodology, I want to bring another scenario. In especially in emergency, what happened, and this is not a scenario, rather this is a real world experience. A lung specialist, he's in emergency. He sees a patient who may or may not have pneumonia. Based on his expertise, he diagnosed pneumonia. But after 2 hours, when clinicians get the radiologist report, he said there's no pneumonia. So because of that delay of the final interpretation, he already gave the antibiotics to patient, or vice versa. So this delay and miscoordination is something which we can reduce by using this the Palmodip. Now coming to the methodology, I mean this is our the flowchart step by step how we developed this tool, how we took the data, how we train it, and there are different components of training. I mean, cleaning the data, training the models, selecting which model is going to work, their evaluation, and differently their testing. So this is our architecture that is only up to building MVP. The further we are working on it, and I can take that at the last slide. Can you please go to the next slide? So here, our solution, Palmodip, is this is how the flowchart is in any hospital setting. Uh, if any patient sees any patient has any lung issues or lung related problem, he don't know what the problem is. He get the X-ray, and then the X-ray can instantly reviewed or diagnosed by this tool, and it will help to at least triage. If something is really worried, let's say there's a possibility of cancer, or there's a possibility of tuberculosis or any infection, that patient can be taken care very I mean in as at a emergency level. If someone don't have that much urgency, that not need to go. So it will, first of all, it definitely reduces the misdiagnosis, help in triaging, reduce the wait time in hospital, and finally it will help to minimize the footfall in hospital. Along with that, so these are the side the benefits or the advantages in terms of in the prospects of clinician and patient. The second one is the whole system is collecting data, and that data is going to be connected with the hospital's workflow, and that data is already diagnosed, labeled, and can be used by hospital for the further research or development, or even that hospital can share that data to pharmaceutical industries, and that can be monetized. The last benefit is, let's say there's a chances of some pandemic or endemic. So this data will give us an alarm. So this could be help as alarming information, or this data will give an intelligence. Oh, something is going to happen in this particular hospital area. So that could be our further extension of getting some alarm of any endemic or so forth. So I shared a demo link to the organizer. If you see there's a time, that demo link is on, I think it's around 60 seconds. So you can play. If not, I can take this later. And there are different components I want to share, but that I can take during fireside chat and question answers regarding what is our next step in terms of testing, validations, how I'm working, what are my plan, that I can take in the during the question hour session.
predict lung diseases early and accurately using advanced AI technology. It collects anonymized patient data, analyzes it with advanced AI, and delivers detailed disease reports and personalized recommendations through a user-friendly interface. Pulmodeep is designed to detect and monitor a range of pulmonary conditions. It is easy to use. Let us walk through how it works. First, go to the home page and select the prediction model you need. Browse your local files and select the X-ray image you want to analyze. Then, simply upload the image. AI algorithm analyzes the image to provide you with accurate results. You'll receive a detailed report with in-depth analysis. Visit DigiBiomics for more information. Take control of your lung health today. Thank you.
Thank you. Thank you.
So now we're gonna get our Sesan and get you all here, and I've got a couple of questions to ask you, mainly because this is a I think that your questions and your answers can be illuminating. So my first question is kind of transparently direct, which is that how does your company approach the lung in space for users, researchers, and potential products? Like, how do your particular tools in your companies are saying, we're going to put our mark in the lung space, in the breathing space by doing X? I'm going to start with Sesan, and then Meraj, you can continue, you can go.
Yeah, thanks. And that's a great question. Um, our approach is from a cellular context, of course. And the way we are doing that is by uh, building um, segmentation models and tracking models for lung specific cell lines. So, like the cell lines that actually are the fundamental components of your lungs. Uh, we are designing cellular models for segment the foreground from the background, like, microscopy context via imaging, and you want to be able to identify that. The second part, which is the tracking, is when the cells are actually moving around, because lung cancer cells move around a lot. Very interesting. They move around a lot. So as they're moving around, we've also designed a model that can follow each cell in your experiment, and it will track it throughout the experiment from each frame, like from one frame to the other, and it would extract all of the cellular features from it. For example, the area of the cell, the perimeter of the cell, elongation, the speed, the distance, so all of that good stuff, it would extract. And we're doing all of that by building models for lung, for building models for like cells from the lungs. And each of this contributes to help researchers that are working on drugs or compounds to treat like different types of um, respiratory diseases to be able to do foundational research on a cellular level a lot quicker.
Okay, Meraj?
Oh, the same question to me?
Yes.
Oh, yeah, thank you. So I think partly I covered this question, so it is very obvious. Pulmo is pulmonology, and deep is the networking, neural networking. It is for lung. And our purpose is not even in the this is something which is covering most of the lung diseases, so it's even the better saying that this is only for lung. not only lung specific one or two or three disease. So that's the uniqueness of our product, is that we are approaching with AI engine, I can say, not one or two or three or four models. Like a holistic AI engine is capable of identifying lung anomalies, tuberculosis, COPD, lung cancer, fibrosis, ILD. Every disease has a unique feature, and our AI engine is capable of identifying all those features precisely. So that is the uniqueness of this model. But definitely, this is for the lung. This is for the lung specialist, and this is for the radiologists who are working in lung domain. That's one thing. The second thing is, this is not a diagnosis and giving a precise personalized treatment. Rather, it has a very wide open window for research purpose, where we can take this data and differently. There are many options where we can do research at not only the symptomatic level, rather in their mechanism level too. That I can explain in a different way, but here I say I see the huge wide window for research purpose. The third option is the pharmaceutical companies who are working in developing the medicine or drug agent related to the drug to the lung. This tool will be helpful in terms of providing uh, data to them. Even that tool will be helpful in terms of helping to at the CRO level. I mean, when they are running some clinical trials. So this is for lung. This is for lung specialists. This is for the lung researcher. This is for companies who are working in lung domain. This is, yeah, lung. Thank you.
Awesome. So I'm going to stick with Mirage. So I have a particular question for you. Your work focuses on multimodal data sets to encompass the disease from different markers. How do you approach multimodal modeling when sources can come from different areas?
Thank you. This is a very, very good question. And in fact, this is linked to the previous one. As I said, there are many lung anomalies we are taking care. Then for sure, we have to deal with multimodal modeling, because the data is coming from different aspects. So we are differently. We just showed the involvement of X-rays, but this is not the end. This is only the starting. So we are going to use X-ray, HRCT scans, clinical information of those patients differently. Along with that, we will be also including genomic data, because there are some lung problems that has some genetics or association. So we will be going to bring genomics data. There are mutations or the biomarkers which are related to lung cancer. That is the further one. So we are definitely going to take X-rays, HRCT scans, genomics, along with that genomics, the clinical profiling. So, to manage this, there is like technical aspects I can quickly walk through. The first thing is data harmonization. So I don't want to go to detail. There are many aspects, many tools. Our data scientists, data engineer, they know how to harmonize the data. Definitely, that part has been done under the supervision of domain experts like pulmonologist or researcher. Then the next step is feature engineering, where we have to drive the features from all multi-model or diverse data sets. Next is the fusion strategies, where we have to fuse data, and it has like a different stages. If you want to combine the data, whether you want to do a early fusion, late fusion, or intermediate fusion, but that needs to be decided based on the data. The next step is we have to manage the missing data. Let's say we got data from different resources, different modalities, different types of data. Then we have to see where the missings are, and we have to impute or augment those missing things. Then finally, we will go to the multimodal data modeling. And that's a challenging aspect, where we are doing so much R&D, testing all aspects of models, large language model, or even the RAG or CAG. So there are plethora of models and libraries, but which one is going to work? This is a space for research and development, and we have a huge team in terms of the clinical researcher, data scientist, data engineer. And I want to share that one more information which I have not listed in the PowerPoint. This is not only one company, so I have a strong partner who has expertise in data integration, data modeling, and back-end and front-end development, IP consulting, and health analytics AI. The other one who has expertise or a stronghold in business component, connecting the hospitals, and we got a deal to take this tool and deploy in a Canadian hospital. So this is the product of 3-4 big teams that we are working on it. So that's how we go: data harmonization, feature engineering, fusion strategies, missing handling data, missing handling missing data, model validation, and after model validation, we are going to really validate this in real world scenario. As I mentioned, we are going to deploy this tool in a hospital, and definitely all needs collaboration: hospitals, researchers, developers, and business team. So this is something which I explain in a very high level, but providing with more information. But that approach is working for us very well.
Awesome. Sesan, I do have a question for you. So lung cells differ from other cells because they need flexibility to expand and contract. How do you solve the issue of imaging cells that can change while making it real time?
Yeah, thank you. And that's another um, very interesting question. Um, you are right that lung cells do have this property, but the way we address that is by setting a protocol for seeding cells. So, depending on your well size, right, we don't want to make that too crowded. So, we want to give the cells enough space to sort of like move around. So, you want to make sure that the density you have in your well isn't overwhelming. So, you don't want to have like a very confluent um, a cell too quickly. So that way you're not going to actually see the dynamic properties of the cells because that's essentially what you're concerned about. And because the media that you're going to be using for your cell culture will differ depending on what type of research you're doing. It could be F12, it could be DMEM, it could be NEM. We typically have sort of like a period where we're testing out the media, the components of the media, and see how much that affects the cell growth. So essentially, we would target the seeding density protocol to a way where there's enough room for all of the cells to move around, and even when they do um, uh, have like some contact dynamics, there's enough room for each cell to grow or to move away as it really wants to.
Awesome. Awesome. So now I'm going to turn the questions to the audience. So, so audience members, I wanted to ask you all, if you have any questions, if you want to raise your hand and they can field the questions or answers, or you could type it in the chat.
I can ask a question. I have in the meantime.
Okay, go ahead.
So I mean, definitely this is for the lung cell lines. So there are different lung cell lines. So how many cell lines you have tested? That's one thing. The second thing, have you used any the ex vivo cells? Cell line, one question. The other one is, do you have a chance to get the lung tissue and extracted cells and then test it? Yeah, this is to.
I'm sorry,
Yeah.
I didn't hear the second question. Can you repeat that, please?
Yeah, so the first question is, there are different cell lines for lung, and even in that case, there are cell lines which will go for lung cancer or a normal lung. So how many cell lines you have tested by this imaging system? The second question is, have you done any testing where you took the lung tissue and extracted the cell lines and tested?
Yes. So, first question, I will just repeat it because I think I have like shooting network now, and I apologize for that. The first question is how many long cell line have we tested? We've done one so far, which was the example I included in my presentation. That's the small lung cancer A549, and it behaves in very, very strange and interesting ways. Um, and your second question is if we have had any primary lung cell line, right? Meaning, you do like a biopsy from like a,
Mhm.
patient or from an actual lungs.
Yes.
If we work with that kind of primary cell line. Is that your second question? Am I
Yes. Yes.
correct?
Yeah. Primary cell lines.
No, not yet. But we are, yeah, we're not, but we actually have a client that's working with in Washington State, Seattle, and we are in negotiation with them to actually get like a primary cell line. And what a lot of people are actually working with now is not even primary cell lines. A lot of researchers that we've interacted with work with iPS cell lines. You just take whatever cell, could be a skin cell, somatic cell, and you do some genetic rearrangement into induce like an induced pluripotency and change that cell line to whatever cell line you're working with. So a lot of researchers are doing iPS cells for different types of diseases. So they don't actually have to do like, they don't have to go through the rigor or the stress of getting a primary cell line because that could be very challenging.
I got it.
So
Yeah.
thank you. Very interesting question.
Thank you.
Awesome. We actually have a question in the chat. One of the audience members was asking, what is the general drug screening in the pharmaceutical industry is. They're thinking like organoid engineering might give better assessment for new drugs, like what is going on in that area. I think that's teed up towards you, Sesan, but also Miraj, you may want to make comments too.
I can take this question. So this has like two component. The first one is the drug screening. So drug screening itself is a big industry, but with the invention of this high computational power algorithm, machine learning models, and AI, there is a huge potential. And in fact, we have done this. So what we did, I'll give an example so it will be much easy to explain. So we know that there is an enzyme is called as glutaminase. Glutaminase is responsible for cancer progression. It's very clear, very straightforward. So if we design a drug or compound that will go and bind to glutaminase, so then we can stop the cancer progression. It's clear. So what we did, we studied the properties, features, structure, 3D, 2D, all sorts of glutaminase enzyme, and it's available publicly. And then we developed our models. Those models, by taking the structure of glutaminase, they screen all the libraries which are available so far in public domain, and they brought us 5 compound, and those compound are based on the algorithm and AI screening. They're supposed to bind and stop the glutaminase activity. This is clear. So now, so those compounds are there, but definitely their physical properties, chemical interactions, the binding domain, everything has been studied. And now those compounds are there, we can test it, and or we can sell to any pharmaceutical industry to test in, as he mentioned, we can test in cell lines, we can test in iPS cell, or we can sell in primary cell lines. So this screening process is minimized, and in traditionally this takes like 3 to 5 years, but screening we can do within 1 month or 2 months. So there's a huge potential in terms of drug screening. The second question is organoid and tissue engineering. That's altogether different domain. in that case, I don't think there is a AI component which will help, but organoid development it's a very challenging process that starts from iPSC. Then you have to bring the different cytokines, different component, and along with that genetic engineering, CRISPR-Cas, CAR-T. It's I mean this question is looks like I said, but it needs a huge complex collaboration with different expertise. So and that's why the organoid and tissue engineering is happening last 30-20 years, but there's no any progress so far. So I think this is helpful, but if not, then please let me know. I can try to explain again. You are mute.
Sorry for that. So, yeah, with that being said, we are at time, but before we do, I have one more question, which is very blunt, but it is the beginning of the year, we have New Year's resolutions. So what are you two both expecting to accomplish this next year? I'm going to say keep it into 1 minute. So, Miraj, you're first.
Thank you. I was actually about to touch that point, but I was waiting for this question, the right time. So as I mentioned, our MVP is ready. DigiBiomics have two arms, the partner arms, Healthlytics, London Health Partner, and they are helping to take this to the hospital. Because unless we validate in a hospital setting, no one is going to believe, and definitely no one is going to use it. So within quarter first, our timeline is to take this tool, integrate there. Integration itself is a process, big process that needs a substantial time. Then we start validating. Let's say then the next quarter we can validate. Let's say we based on the frequency of X-rays. We will, for example, just I'm throwing a number. We validated with 1,000 data points. So that will give us a edge that based on that we can go to the market, start soliciting and say our tool is ready to deploy or to in any other hospital or to the next level for the commercialization. So our target is Q1, Q2, Q3 maximum. We will bring this product in a real world so that we can start helping community.
Awesome. Sesan, please wrap us up.
Yeah, thank you so much. Our plans for 2025 is to onboard more researchers. Our mission and our value is to help cellular biologists build a lot faster. And we do this by collecting and training a lot more models for specific type of diseases. And our goal is to be able to train our models for lung diseases or lungs related diseases on top of what we have, get a lot more users to use a platform for extracting cellular features from their experiments in a seamless way. And I hope that we would have potential users in the crowd today. I can't wait for you guys to try out a platform, and I hope we get a lot of feedback on what we do. So our goal is to onboard a lot more researchers this year, build a lot more models for different types of diseases, and have a pipeline where anyone in the world that's working on any kind of research can just easily plug in using a web link and have access to powerful model that would drive their pharmacy research.
Thank you very much. I want to thank the speakers for both coming today and giving us some very much knowledge to not only think about, but very innovative based products and research that's going to really, contribute to the space of the lung space, especially in the biotech tech sector. I will be sending out ways in which you can contact them so that you can be able to reach out to them. Please watch it through the Luma post because you'll see how they'll be able to contact them. Thank you very much. We hope you have a great day. We hope to have something in the near future where we have more people in different areas that you can learn from and grow from there. Thank you very much and have a great day, everyone.
Thank you,
Thank
everyone.
you.
Thanks.
Bye.
Bye-bye.